go. First thing that we did today was to look at our streak plates from last week. And the question we're asking, do we have isolation, right? But what was the purpose? Why do we want isolated colonies? What should those hopefully maybe be? Pure cultures, right? That's the important term, right? Okay. But we're looking for a single species, right? A single species of organism. And so this particular method, and there are other methods out there that we use, the streak plate method, was to hopefully spread them out so you had a single cell or cells of the same species and they grew up. Now we had an interesting phenomenon due to our constraints in lab right now. One of the organisms that I gave you, I think I told you a little bit about last time, it's this reddish one. And it's red, it produces this pigment at cooler temperatures. So when we grow it at 25 degrees Celsius, it produces that pigment. When you grow it at a higher temperature like 37, equivalent to body temperature, it actually does not produce that pigmentation. That heat must shut down that mechanism, and so they're actually white. Or you could almost say colorless. But when you guys look at your cultures, you've got polka dots, don't you? Right? And so, especially if you picked a pink culture, you'll notice that, you know, it's kind of pink in the center and the edges are kind of white. This is an interesting phenomenon that has happened to me more than once, thanks to Delgado. They were working on the air conditioning system this weekend. So they shut off the air conditioner in here over the holiday weekend. So those incubators do not have cooling elements to them. They just heat. So it got too, what in here? Too hot. So it was probably pretty darn hot because it inhibited their growth. But the cool phenomenon about this, always a great teaching moment, is that where do you think the newer growth is? When you look at that, look at your colonies or look at one of your classmates. It's on the outside. Why? Why do you think it's on the outside? It started on the inside and it spreads out, right? That's how they grow. So it kind of gives you an idea of how they grow on these plates. They even stack up on each other. But growth is always going to be greatest, and the newer growth is always going to be on your edges. And this is because there's more nutrients available and less cells, right? As they grow up, they're stacked on top of each other, right? And it's less, it's not as easy to acquire the nutrients from the food source. Make sense? So I might steal one of those and take a picture of it for next time. Not an easy thing to replicate. So typically we would grow, say, at 37 degrees for about 24 hours. And usually what we would do is put them in the refrigerator even to slow growth. Especially for some of you guys that have a lot of growth and colonies kind of ran into each other. That wouldn't have happened. The problem is that we left them incubating for a whole week. Right? So this is much longer than you traditionally would do. That's okay, right? It's just we're teaching. Right? We're not doing scientific experiments where you've got to have that pure culture that we got to pick off that plate and run tests on. But a plate is a totally different environment from, say, a broth tube. And we've got some broth tubes, but one of the things that we don't have in this lab is what's called a shaking incubator. And what that does is it shakes the tubes, and so especially organisms that need oxygen, it aerates them. It also mixes them up so that there's a pretty even distribution of food and waste within that tube. And so you tend to get the most uniform, the best growth in a broth culture because of the different architecture that the organisms are living in. Now some of them are modal, so they can swim around on their own, and that really kind of helps them out. So definitely, again, today, when you get your cultures, look about where the growth is in those tubes, right? And what it looks like on the slant. Make a note of what you see, okay? I quickly looked at your cultures. I think everybody has growth. If you don't have growth, use someone else's culture. Ask to borrow someone else's culture to do your slides. Sometimes they don't behave, you know, or sometimes we don't do something quite right. It happens. But so under those ideal conditions of being in a broth and being shooken up, organism like E. coli could, under our ideal conditions, double itself every 20 minutes. So we could calculate, right, over that time frame, if you started with a single cell, about how many cells would be in those colonies, right? So that colony, the reason why you can see it is because it is a lot of bacteria. Theoretically, and we're not going to do out the math, right? Theoretically, but it's here if you want to know it, it would be 1,000,000 billion bacteria, right? For this one example, for E. coli, right, that can replicate potentially as quick as every 20 minutes. And here we're actually only calculating every half hour, right? We didn't even do it based on 20 minutes, right? So 2.8 times 10 to the 14th. Imagine that many zeros, right? We're talking about a lot of bacteria. That's why, too, when we're taking our organisms and moving from culture to culture, I tell you guys, you just need a little bit, right? And I'll say the same thing today when we're getting ready to make our slides, right? You don't need to scrape the whole surface of the slant. You'll have way too many bacteria and make it difficult to stain and visualize them under the microscope. So there have been studies, right? And we have reference to one in here. And as we were saying a moment ago, you know, it's going to be different, a broth culture, as opposed to when we grow them on the Petri dish, when they're on the agar, because especially if they're non-modal, and in that environment, it's not going to be easy to swim, right? Some of them kind of wiggle along the surface, right? But they're going to be confined by the constraints of that type of environment, especially, you know, when it comes to availability of oxygen, right? If they're piled up on each other and nutrients become less available. So one of the things that we're going to talk about later in the course is, you know, organisms that can grow without oxygen, which we don't mess with in this course. Ones that are strict anaerobes that cannot live in the presence of oxygen, because it's really hard to create that type of environment. We will work with some that can grow without oxygen, that are anaerobic, but can also grow with oxygen. So all the ones that we're going to work with can tolerate oxygen really well, or can grow really well in the presence of oxygen. This is the easiest environment for us to replicate in the lab. So our facultative anaerobes can grow anaerobically, but they can also grow aerobically, and they're going to grow better with oxygen, right? But they'll still grow without it. So depending on the organism and the organism abilities, right, within the confines of that type of media, right, you'll see the different growth patterns. And so one of the great things about the phenomenon that happened to us, you can really see that that's probably the newer growth on the edges of that colony. It's really kind of cool. So this is a link to a particular article. They estimated for E. coli that they actually, in fact, under those conditions on a plate, actually only about 1 billion times are they going to replicate, right? And that's about a 3.3 billion bacteria in a colony. But again, different organisms grow at different rates, right? And the conditions of that growth really are going to affect how many organisms you end up with. Take-home message, lots of bacteria, right? Lots of organisms when we could see them on those plates. So can we experimentally determine this? We can. And there are several different methods. And later on in the semester, this is a little preview into some of the things that we've tried out in the lab. Different ways where you could take a colony or a set of colonies and actually grow them up and determine about how many were in that colony. The problem with that is when you grow the organisms, only the viable living ones will grow. So if you had some dead ones, you wouldn't be able to count them. So we're going to do a technique after midterm, literally after the midterm exam, where we're going to dilute out a sample of bacteria and try and estimate or calculate how many organisms we had to start with. Right? So again, to give you an idea of this very large number of organisms that can be present. You know, when we have those really turbid broth cultures, right, where it's really cloudy, you're talking about millions upon billions of cells. Right? A lot of bacteria. No drinking the cultures, no licking the Petri dishes, right? Some of these are pathogenic. I'll kick you out of the lab. All right, so for today's lab, we're going to stain, and we're going to use two very simplistic stains. One is a simple stain that's most commonly used using what we call basic dyes, and then we're going to do negative stain. And you're going to kind of do a combination of this when you do the capsule stain next week. So it'll make sense, right, as we talk about what each one of these does, how we can put them together to achieve the goal of a capsule stain. So I'm not going to go over each one of these terms, right? We've just been starting to put them in the PowerPoint so you guys have a nice glossary to quickly refer to for terminology. As it relates to this terminology, it's in your book. It's kind of confusing, right? Peter and I agree that it really isn't all that important which part is the color and which part is charged of these dyes. The important thing is that the dyes contribute color, and why and how they interact with your cells is dependent on what charge they have, right? So these terms, I promise you, I will not ask you them. But they're in your book, and they're defined for you there. So here's two examples of two dyes, and it's like right down to the molecular structure. Okay? Do you see what's different? Not much, huh? To really look, it's like one of those find what's different. Anybody find it yet? It's at the top of the crystal violet. It's almost like its little antennae there. That's the only difference between these two molecules. But yet you have two very different colors, right? And Peter loves chemistry, so he explains it right down to the chemical level of how electrons and everything move. I'm not going to get into that for you, but if you're interested, it's here, right? So it deals with what we call resonance, okay, and how the electrons move, right? So the Erica simplistic version, right, is how the electrons flow through and interact with these molecules is going to determine the different colors that they emit. And so for this guy, it actually absorbs in the red range, but then it's going to emit opposite from that, and so it produces green. And so this also kind of equates to why plants are green, why most leaves are green. It's actually, they're not absorbing the green light. They're absorbing the other lights, and the green light gets bounced back to you, right? And so that's why it appears green. When it's using other pigments that absorb different waves of length, that's why it changes color, right? And that's because those wavelengths are different at different times of the year. So it's part of the reason why you see the color change that happens in the leaves. They're using different pigments due to the different light availability during the different seasons. And then there's some of them that just go through that change anyways, so we even still have that here, even though probably the light isn't really the factor. Then you have the whole snow problem, right? Having those big leaves, not a good idea. So they go into their dormancy period. So for crystal violet, this guy absorbs over here, so then emits the violet or purplish color. We use this dye, and we'll use this dye today just by itself, but we use it in combination with another dye when we do gram staining. So we have the two different colors in that differential staining procedure where you can differentiate one group from another. Malachite green we'll use to stain our endospores when we do the endospore staining. Sneeze. Okay, so microscopy helps us accomplish three very important things compared to just using our eyes, right? Why do we use a microscope? Well, one, you're not going to see anything, and if you are, you're not going to be able to resolve it and see it really clearly. And we can calculate this right down to the math for our microscopes, right? And this is a formula you need to know, right? So it's what on the top? Wavelength of light, right? And we use special filters to use particular wavelengths. And what's on the bottom of our equation for limit of resolution? Numerical aperture are two important lenses. Which ones focus light on your microscope? Which is the one that focuses it underneath your slide? Condenser. Which is the one that's going to focus the light so you can actually see your image? Above your slide, your objective, right? Those are the two important lenses that comes to focusing light and actually seeing your image. The numerical aperture is like its prescription, right? It's how good it's able to focus that light and give you a nice, clear image. And we actually get a number, right, which is a distance. And that is the minimum distance that has to be between those two for you to dissolve them as two separate. If they're too close, what's going to happen? They're going to blur together, right? So we want to see things clearly, right? So those lenses, we're We're limited when we're using light, though, right? Because we have to stay within the visible light spectrum, right? But we can calculate this, right? And this is a formula you do need to know and be able to calculate. The other is magnification, and again, this is due to the lenses, but two different sets of lenses. Which two actually magnify the specimen's image? Objective and ocular, right? The ocular does not change on your microscope. The objectives, right, you have choices. So how do you figure out total magnification? Multiply the two, right? Your equation is, right, objective times condenser. Right? The X is the units, right? It means how many times magnified. What's the units for limit of resolution? Yep, nanometers, and it comes from the light. The third thing is contrast. Some microscopes can help create contrast, right? Others, we have to do other things to create contrast. So what do? We stain, right? We stain them, okay? The problem is, sometimes when you stain them, what do you do to them? You kill them, right? Most stains are toxic, okay? So if you wanted to see them move, if you kill them, are you going to see them move? No. Right? So this creates problems, right? Especially later on when we want to see them move, but we can't stain them to help us see them, so we have to, use other methods to help us visualize them. So there's several different types of microscopy that utilize light. Which one do we use in lab? Bright field, right? Where the background is bright, right? And the specimen itself absorbs the light, so it has to be colored, right? Whether we stain it or whether it's naturally pigmented. Dark field microscopy is the opposite. The background is dark and the organism is illuminated. There are several ways to achieve that, right? We kind of do a version of that when we really decrease the light later, when we're trying to observe motility of the bacteria without staining them, right? We try to make it as dark as possible so that just the cell is illuminated. Phase contrast uses special filters and knocks the wavelengths of light out of phase with each other. And in that, it creates contrast. So the nice thing about that type of microscope is that the microscope itself creates contrast. So if the organisms aren't very easily distinguishable from their environment, if they're very translucent like most bacteria are, it helps us in the visualization of them. We don't, unfortunately, have one of those microscopes yet, but I'm hoping. Fluorescent microscopy is widely used in microbiology nowadays. We a lot of times use specific dyes that have fluorescent ability. This uses ultraviolet light to cause them to fluoresce, so it has to have special filters on it so you don't damage your eyes. And some organisms are naturally fluorescent, but more oftenly we're dyeing them with specific dyes that can actually even bind to specific components of cells for help us visualize it. So stains help provide contrast, right? The problem is it will kill them, right? So when it comes to motility, we can't stain them. Stains can be applied broadly to determine, and that's what we're going to do today, is just shape and arrangement, right? You'll see the individual cell shape, and if they stay together in groupings, what we call arrangements. Or they can be used to look for specific structures or characteristics. So the types of stains that we'll do is simple and negative staining today, which is, again, just morphology, just shape and arrangement, is all you're going to be able to tell about those organisms. Gram stain and acid-fast stains are to specialize what we call differential stains, because you get one of two results. You can differentiate one group from another. And these are based on specifics of their cell walls, is why they will stain differently in this procedure. And also why we'll use two colored dyes to help us differentiate the two different groups. For endospore and capsule staining, these are specially designed ones to be able to see specific structures. The endospore is an internal structure, hence the endo component, and the capsule is an exterior structure. And again, we have to employ particular methods to the staining to be able to visualize these structures. Right? So those staining procedures are specifically designed to be able to visualize these structures if present. Right? Not all organisms have endospores or capsules. All right, so they're what we call differential, right? They're going to help us differentiate one thing from another, whether it be a structure or a cell wall composition. But in addition, right, since we are going to be staining them, you could also be able to tell the shape and arrangement, right? It's just going to tell you more information past just shape and arrangement. So these are the common shapes and arrangements and the terminology that go along with it. So the first one, remember that these are three-dimensional organisms, right? So this is spherical, like a ball, if you were to look at this organism. And the term that we use for this is coccus. Cocci, if it's plural, if you're talking about all of them. A single one is a coccus. Multiple ones are cocci. This is Latin. For me, how I remember, it's kind of the opposite of what I'm used to. I is singular, right, in our language. For them, it's plural. For us, is plural, right, meaning many. For them, it is singular, meaning just one. So for the next one, these actually form a characteristic arrangement where they stay together in pairs after they divide. So it's Diplococcus if you're talking about just one pair. If you're talking about all the pairs you see in this picture, it would be Diplococci. Right, so that's the difference there. It would be Diplococci, right? Switch out the I for the US. The next one, you'll notice that there are 4 cells together in a square grouping. This is referred to as a tetrad or tetrads for plural, the usual plural we're used to, just add an S. Right? And one of the organisms we work with today has this characteristic arrangement, so hopefully you will see this very easily under the microscope today. It's one of my favorite ones. It's kind of pretty. The next one, notice again they're cocci, so they include that in the name of this arrangement. And then also they're forming a chain, right? And the prefix for chain is strepto. Streptococcus prurigens causes strep throat. So one of the ways I help differentiate strepto from another one that people commonly mess it up with is staphylo, that we'll get to in a moment. I always think strepto, I think strep, I think about strep throat, think about how sore. I don't care how long ago it was, everyone for the most part has experienced strep throat, right? Your throat is so sore you don't want to swallow anything. Well, imagine trying to swallow a chain. Forget about it, right? So I think about my throat, that tube. I think about, not wanting to swallow anything, including definitely not a chain, right? So strepto, I think chain. I think about my throat. I think about how I wouldn't want to swallow that, right? These little stories we tell ourselves to remember these things. Ever since I started doing that, I never get those two mixed up. So the next one is a packet of 8 cells that forms a cube. And this is Sarcinia. And this is also a genus name, just like strepto is. And as you can imagine, the organisms in that group were named after their particular arrangements. The next one is Staphylococcus. If you're talking about just one cluster, if you're talking about lots of clusters, it's Staphylococci. Again, one of the organisms we're going to work with today is going to have this characteristic clumping, right, or clustering effect. And again, a lot of times we abbreviate the name of the causative agent for the infection when it comes to staph infections. They're referring to a Staphylococcus, probably aureus, that can cause really bad skin infections. A lot of people actually have this organism growing in their nasal passages. It's when it gets into wounds and stuff like that it can be problematic. And then especially if you have a strain that's antibiotic resistant. So most people say MRSA, but it's actually an acronym. It's MRSA. It stands for Myctoline-resistant Staphylococcus aureus. So this is a particular strain of staph that is resistant to penicillin-like drugs. Right? So that's the bad news about that, is that we're limited in what we can use to treat it because it's resistant to one of the common antibiotics that have been used in the past. The bad news is that now we have vancomycin-resistant Staphylococcus aureus, because that was the next widely used antibiotic for those types of infections. So it's an ever-ending struggle. The problem is the bacteria probably outwin us. Because remember, antibiotics are stolen technology we stole from other microorganisms. Right? So unless we steal some more technology or invent some more technology, they might eventually beat us if we're not careful. So the next one is commonly, as I said, referred to as rod, but the actual term would be bacillus or bacilli. And the best way I can think of for describing, three-dimensionally, this would be like a sausage or a hot dog, right? If you like candy, Mike and Ike's. Hot tamales are my favorite, right? That shape, right? So you can imagine that particular shape. And then, of course, if they form pairs, we already learned that the prefix for pairs is diplo, but these are rod or bacillus shapes, so it would be diplobacillus. And I have to say, I've never seen one. This next one we will see hopefully today if we get enough cells. And so it's a chain of bacillus, so it would be strepto, not staph, strep, chain, strep, thick of your throat, right? So streptobacillus or streptobacilli if you're seeing several chains of them. Yeah, this is pretty much where it ends for this lab. As far as what you will see under the microscope is basically chains, rods, and cocci, right? Bacillus and cocci. That's all we have. In different arrangements, right? But that's it. That's as exciting as it gets in microbiology lab. We don't have any of these other cool shapes, mostly because they're difficult to grow, and then of course there's a lot of really bad pathogens in these groups. So in the next couple of slides, I'm going to give you guys some examples. Again, this is not anything I'm going to quiz or test you on. Especially you guys going into allied health, it's good to know about different pathogenic organisms and what they cause and how they make you sick. So this next one is still kind of a rod shape. It's not very well shown in this picture, but it has a curvature to it. And then also in this picture, it actually shows that this organism has flagella. These little lines are representing flagella. But the shape of being a rod with a little bit of a curve to is referred to as Vibrio, which is, again, a genus name. You've probably heard this name before, right? The organisms in this genus have this characteristic shape. A lot of them are also motile by flagella. The next one is a much longer rod that has several curvatures to it, and that would be Spirilium, or Spirilia, add an IA if it's plural. This picture also shows flagella, in fact, multiple flagella, those lines coming off both ends. This is all the same organism, but you don't see a uniform, constant shape repeated over and over again. This phenomenon is referred to as pleomorphism, or pleomorphic, and this is where they don't hold a characteristic shape. And there's a whole genus of bacteria called Mycoplasmas that don't hold a particular shape, and the reason for that is they're missing or lacking a cell wall. They have a cell membrane, but they don't have a cell wall, right, like most bacteria do. They're still bacteria. They have other characteristics that put them in that category, right, like not having a nucleus, which is a characteristic of what cell type? You were right the first time. Don't second guess yourself. Eukaryotes, right? You meaning true, kernel, right? They've got it. All these bacteria, no nucleus. All prokaryotic, right? Lacking a nucleus. So, and then the last we have are those spirochetes. I showed you that video that I have a link to in my course. And these guys are actually helical, right? And they just look like little ziggly lines under the microscope as you see here. But in three-dimensional space, they're actually like a spiral staircase or a slinky. And these guys have that unique ability of being able to corkscrew through their environment. And the reason for that is they have something like flagella, but they're called axil filaments, and they run along the outside of the cell in an outer sheath. And so this is the cross section here, and this is them running along. And this allows them to hold that characteristic shape and to be able to corkscrew through their environment. So as I said earlier, both syphilis and Lyme disease are caused by organisms that are spirochetes, that have this shape and this ability. And that's actually one of the reasons why sometimes someone, when they get bit with a tick that's infected with this bacteria that causes Lyme disease, they'll get what's called a characteristic bullseye rash. And that's because the bacteria swim out from the bite wound 360 degrees, and then the immune system responds and tries to kill them, and that's the rash that you see. But that's why it's circular, because they can swim out from where they were injected into the skin. Doesn't always happen. That's a pretty good dead giveaway that you have that particular type of infection. The good news here in Louisiana, this bacteria is very rare, extremely rare, where it's much more prevalent in other places, especially in the Northeast. It's actually why it's called Lyme disease, because it was discovered in Lyme, Connecticut. So, yeah. Syphilis, on the other hand, is here. Right? And you can get it, and most of us know how you can get it, right? It's a sexually transmitted disease, right? So, huh? Oh, no. The good news is it doesn't corkscrew through your skin, right? These organisms, even though they can corkscrew, can't penetrate your skin. Your skin is a very good barrier, so it would have to get in another way. But then once in your body, the softer tissues, it can corkscrew. Good point, though. But yeah, there's very few organisms actually can penetrate your skin on their own, which is good. Your skin is a wonderful barrier when it's not breached, right? It's when you have wounds that then you're susceptible, or when something bites you like a tick and injects stuff into you. So we always talk about the bad stuff when it comes to bacteria, right? So Mr. Hickman actually found and saw this interesting article on a study they were doing on a tribe. I forget exactly where these guys are, where they're much healthier than we are, and you see them there rolling around in the mud, and you're like, oh, really? But we actually, to a certain extent, in developing countries, we almost become too clean, right? We limit our exposure to microorganisms, which limits our exposure to the good ones, right? They're good and they're bad, and our fear of the bad is really kind of put us at a disadvantage, and we're not as well exposed to other microorganisms in our environment. So they have a very diverse microflora associated with their gut and with their skin, that some of them, don't even experience some of the diseases that we experience in the developing countries. So very interesting level of study. I always joke with my friends, because they're like, the courses that I teach are like, how do you not like agoraphobe? How do you not be cleaning all the time? And I said, when I was a kid, I ate mud pies, and if I didn't live in New Orleans where there could potentially be lead paint in my yard, I'd let my kid eat mud pies too. Because it would probably be a good thing to be exposed to the microorganisms within his environment. Not to say that I will go out and do it tomorrow. Like I said, I know for a fact there's lead paint in my yard. Okay, but being exposed to certain things aren't necessarily a bad thing, right? But when we know we've been potentially exposed to something bad, being clean is a good thing. Obviously, washing your hands after going to the bathroom, right? There is the potential. There are microorganisms in your gut. And if they're not reintroduced back into your gut system, if they get someplace else, like into a wound, they can create problems. Right. So a little note, interesting fun facts about good bacteria. They're not all bad, but then we jump back into the bad and somewhat bad. So Streptococcus epidermis is one of the ones we'll work with even today. It gets its name obviously because it's commonly associated with our skin. This isn't the one that really causes the staph infections. It's the Staphylococcus aureus that can be pathogenic. So there's some fun facts for you guys on that one. Streptococci, as I mentioned, Streptococcus pyogenes is the one that causes strep throat. This is a pretty mean bug, can cause other serious complications like scarlet fever when it produces a particular toxin. Streptococcus pneumoniae is another bad one that can cause serious pneumonia, but when it spreads to other areas, it can cause things like meningitis and some pretty bad stuff. Vibrios, Vibrio cholera is one that can cause serious problems. This is one commonly when water supplies get contaminated with fecal matter that may have this pathogen in it. This is one that we sometimes are concerned with here in Louisiana if you eat filter feeder shellfish like oysters. But come to find out, it actually isn't necessarily usually cholera. That's a little bit more rare. It's more likely this Vibrio, which can cause some serious problems as well. But this is why when you eat raw shellfish, they have that warning that it could be dangerous to your health, right? If you go to any seafood restaurants where they sell, they usually have that warning there. It's pretty highly regulated. I eat raw oysters all the time, right? I run the chance, right? But, you know, it's good to be educated as to what you may be exposed to, right? And the important thing is to seek treatment. So I had a student here that traveled someplace else and doing some field work, and she got Lyme disease. And she's originally from the Northeast, so she felt horrible, and then she found out she had this rash, and she took a picture of it, sent it to her mom. She's like, "You have Lyme disease. It's that characteristic bullseye rash. You need to go to the doctor." And she, of course, had to explain to her doctor. She had to have her blood sample sent to Tulane Tropical Medicine here. She's like, "It's not that rare." Right? But, you know, they're not used to experiencing that here, right? You know, it's in other parts of the country. So, you know, that's one of the main things that people don't think about is travel, right? And what you may be exposed to, even within this country, never mind going to other countries, and you need to be aware of that. One of my professors, same thing, went away to another country. He came back with an infection. He goes to the doctor and he's like, "I think it might be this. I've been to this country and these are the symptoms." And they looked it up together, like, sure enough, this is what you've got, right? So your doctors can only do so much if you tell them the information that they need to know too, right? Which isn't always the easiest thing. You know, when you asked me what I had to eat 2 days ago, I'm like, I don't know. I don't know. Did I eat something bad? Did I go out to eat? I don't know. So yeah, that's as exciting as it gets, but it's pretty cool. Especially when you achieve something, right? And you get your microscope focused and you're like, "Oh, look at that." And of course you'll hear me say beautiful all the time because I just think they're beautiful. But they are. So we're going to see two different representations today. We're going to see where the organisms are going to be stained. So what shape are these again? Bacillus, or commonly just referred to as rods. Here, these are what shape? These are the tetrads, right? And the individual cells are cocci, right? They're little circles, but you'll notice that there you get that characteristic grouping of 4. So that's a tetrad. And you will see, hopefully, a slide that looks just like this today. Okay? And a slide similar to this today. So how does this all work? Well, bacterial cells have an overall negative charge. And we're going to go down to the molecular level and see how that is. There's 2 different types of stains that we use. Basic dyes and acidic dyes. Basic dyes that we utilize in this lab are crystal violet, methylene blue, and safranin. And as you can tell by their names, but also the coloring here, safranin happens to be a red dye, methylene blue is blue, and crystal violet is purple or violet. All of these guys have an overall positive charge. When they're in solution, they're going to give up their hydrogen ions. Excuse me. They're basic. They absorb hydrogen ions. They become positively charged. Almost got that one backwards. So they're going to become positively charged, right? They're going to bind to the hydrogen ions in solution. What do we know about opposites? They attract, right? So in this case, because the cells are negatively charged and the dye is positively charged, they're attracted to each other. So the dye literally goes into the cells and it's going to fill them up like a water balloon. For negative staining, on the other hand, these are acidic dyes. And some of the ones we'll use are Congo red. Eosin we'll talk about, but we don't actually use. And nigrazine we will use today. As you can see, eosin is kind of a reddish color or pinkish, and nigrazine is kind of a black grayish color. These guys, it's the opposite. These guys are going to be negatively charged, and that's because they're going to lose the hydrogen ions, right? They donate hydrogens to the solution, So they become negatively charged. So now we have negative cell, negative dye. What happens? They repel each other. Like charges are going to repel each other. So when you ever take two magnets and you try to put them together the wrong way, that's because you're putting the two like sides together, because it is a polar molecule. So they'll just keep flying apart. So that's what's happening here, is the cells themselves are resisting the dye. So the dye stays outside the cells and stains the background. And these are going to be your easiest slides to focus, because you'll have all this background to focus on. So go in order as I have it for you guys' procedure today, because it'll help you. So what makes the cells negative? Anyone know what molecule this is? You got a nitrogenous base, a nucleotide. It's a sugar and a phosphate. Nucleotides are things like A's, G's, T's, and C's. Nucleic acids like DNA and RNA, right? The phosphate groups that form that sugar phosphate backbone are highly negatively charged molecules. So that's one of the attractants, right? And we've got a lot of nucleic acid inside of our cells. And then the cell membrane itself is made of phospholipids, which those phosphates too, really greedy oxygen here, pulls most of the electrons towards it. And that's why that part interacts with the water either outside of the cell or inside of the cell. And the fatty acids, right, of that lipid bilayer go together because they don't like water. Right? They're water fearing. But all cells, even our cells, are highly negative charged because of the phospholipids and the nucleic acids, and even some of your proteins are highly negatively charged. So what's the consequences of leaving this in? For simple staining, so we're using a basic dye, right? The dye is going to go rushing into the cell. What happens if you leave that dye on too long? It's going to enlarge it, right, and distort it, and even cause the cells to burst, right? So we don't want to overstain. So my golden rule in microbiology is 1 minute. If you're not sure, do it for a minute. Same thing for gram staining, right? Some procedures out there will have a bunch of different times and seconds. Just do everything for a minute, right? It's long enough, but not too long. And it's easy to keep track of on our clock, right? Just watch the second hand go around one time. So that's what we will do today, 1 minute. And most of the time, unless it calls for longer staining times like the endospore and acid fast, we do 1 minute. And so again, and they would swell them up. So especially you have diplococci or staphylococci or tetrads, if they're really swollen, you wouldn't really get to see that arrangement, right? It'd be really hard to discern how many cells and how are they arranged. So what happens if we don't stain them long enough, or we understain them? What's the problem with that? They're not going to be visible. The whole purpose we're staining them is to create contrast to see them, right? If you don't create enough contrast, you're just not going to see them, right? It's going to be very difficult to see them. Which is the case with some of the lighter dyes and some of the procedures that we do. They just don't stain well. So another important concept is surface to volume ratio. And for the two major shapes that we look at, rods and cocci, and in truth, all the other shapes that we talked about, Spiralium and Spirochetes and Vibrio, they're all just long rods, right? Just with some curvature or spiraling to them. So in essence, most of them you could consider it being a rod or a cocci as far as surface area goes. Now, of course, a rod has kind of rounded edges for the most part, just like a sausage or a good old hot tamales. But for mathematical calculations, we figure it out as if it was a cylinder, right? The good news is this math you don't need to know, right? Just understand the concept, right? But for some people, seeing the mathematical equations, seeing the numbers really helps, right? Understand the concept. The important take home here is that for a rod and a cocci, with equal volumes, the rod is going to have a greater surface area. There's one exception to this where there's a rod shape-wise that has the same surface to volume ratio as a cocci. But for the most part, the rods are going to have a greater surface area to volume. So what, right? Who cares? They got more surface compared to the stuff in the inside. Why is the surface important? We're talking about a cell, aren't we? What is a cell? What defines a cell? What layer? The cytoplasm is the inside. What is the barrier that holds that cytoplasm together? Cell membrane. Every cell's got to have a cell membrane. Some of them have a cell wall that helps with support and shape, as we see here, right? But every cell, to be a cell, has a cell membrane. We say that's a semi-permeable, right? Which means that only some stuff is going in and out. In order for that cell to survive, especially a single-celled organism, what has to happen all the time? Stuff going in and out, right? If you have a large surface area, lots of room for that in and out, right? That exchange with the environment. So it is advantageous to have a greater surface area compared to the volume, the inside stuff you're supporting. Make sense? So that shape is better adapted, right, for that type of exchange that a cell has to do. Right? That's one of the reasons why we talk about it. So, again, lots of math if you're interested in it. The one exception where the two are equal, rare, but it can happen, where there's a cylinder and a sphere that are perfectly matched. But for the most part, the rod is going to win out. Right? And this is the most common ratio, height or length to radius. And so we have some demos, some visuals to help us. Right? So imagine, right, different sizes, right? So different lengths. Right, so it's surface to volume ratio, right, this little guy. This one, if you just take into account the bottom part of this canister, right, that rod portion is about equivalent to this tennis ball. This is our model for representing what arrangement? Streptococcus. Coccus for the individual cell, right? Strepto because it's a chain. This one would be diplo. Good. Okay, so the next thing we're going to do, I'm going to share with Andy, is I want you to take out the Play-Doh and give a piece to each one of your classmates. And here, I'll give you guys some extra. Divide that between two of you guys. Okay, so once you have your piece, what I want you to do, I stole this from my son, he doesn't need it. Leaves it all over my house, and I don't even have a carpet. Yeah, I think I said I stole it. I was like, no, we're done with this stuff. Okay, so I want you to do, once you have your piece, everyone got a piece? Hopefully you don't have a very big piece. You want to divide that piece into two equal pieces. One of them I want you to make into a ball to represent what? Coccus. And then I want you to make sausage to make what? Rod. Bacillus. So you can see, right, if you had two relatively equal sizes of Play-Doh, you would have the same volumes, right? And you can even see greater surface area for your rod, right? You can feel and see this as opposed to your little cocci, or coccus. Bacillus. Okay, and then I'm going to take mine. You can do the same if you want to. I'm going to make it into Vibrio. And then, I'll steal yours. Still a rod, right? Now, I really am, right? Then if I just go like this, say, the first one was Spirochetes, right? It was like a spiral staircase. This is Spirochetes. Right? We're just all right. We're done. Yeah, they're helical, you know. It's slinky even. All right. So some more analogies, right? So which items are going to cook faster? The ones that have a greater surface area as compared to their volume, right? So the heat can penetrate all the way through when it comes to this. So this guy is going to cook faster, right, as opposed to the meatball, right, where look at, there's our cocci, right? Here are some rods, our good old sausage, right? You're going to get better heat distribution, right? These guys are going to have to cook longer, right? Slow heat transfer to that center. Alright, so more math, but take home, right, is that the cocci, you'll notice, are all pretty much the same size, right? Because they're limited, because the larger they get, they're not going to be able to create, they're going to end up with more volume than surface area because of their shape. When it comes to rods, though, they can vary the length as we saw on that other diagram, right, and really create a large surface area for exchange to happen. There is a downfall to that, though. And so, again, just represented in a different way. These guys are stuck, right? Spheres are stuck, right? They can only get so big, right? So, but rods, we can have variation, although typically this is around the average for them, for their surface to volume ratio. Again, it's still above, right, what you're dealing with spheres, right? They have greater surface area as compared to the volume they get to support. So even at the organism level, right, with our good plants, right, they're doing exchange, right, gas exchange that has to happen for photosynthesis to happen. So what do? Some of them have these nice broad leaves, right? They're really thin so that they can do exchange. Even the needles, right? These are better adapted, though, for the loss of water, right, under those conditions, right? They're nature's little solar panels. So what condition where one of these guys is actually going to be at a disadvantage is when it comes to water, right? Water is very important for organisms. And the greater the surface area you're going to have, the more loss of water. So as you saw with those broad leaves, that's one of the things that you're sacrificing as opposed to the needles, is that water loss. So because a rod has a greater surface area in a dry environment, it's going to have a lot of water loss, right? So the cocci in that case outweigh the bacillus because they're going to be able to preserve themselves better because they have less surface area. Make sense? So there's a reason for it because, you're like, well, why isn't everything just rods then, right? If rods are so much better. But there are some conditions where actually being a cocci is better suited, right? If you're in a dry environment, you're going to be able to preserve water, not lose as much because you don't have such a great surface area. But, right, if you don't have to worry about moisture, right? If you're in a nice, warm, moist environment, then who's going to be the better suited?
Rod.
The rod, because they have what? the high surface area compared to their volume that they're supporting, their stuff inside.
Is there a situation where if it's too wet, outside would be best to not expose it too much water?
No, shape wouldn't matter in that case. So he asked the question if there was too much water. Shape wouldn't help or be a deterrent for either one of them. And there is a case where there could technically be too much water. But the added advantage that bacterial cells have is the cell wall helps protect them with osmotic pressure. So that's what would actually help them. Doesn't matter the shape of it, just that they have it. So the next is negative staining. And so when we do negative staining, remember this is using acidic dyes. And so they're repelled by, they're negative, the cell is negative, they're repelled by the cell, they're going to stain the background. So why doesn't a negative stain colorize those cells? It has to do with the fact that the stain is negative, the cell is negative, they repel each other. So given this example, and this is a good lead into some of the stuff we're going to do next week where you combined. So I think in your manual it didn't actually tell you, so you'll notice I added this. So eosin is an acidic stain. So again, this tells you what charge it has, right? What charge would it be if it's acidic?
Negative.
Negative. So what's it going to stain? If it's negatively charged, is it going to stain the cell or the background? The background, right? It's going to be repelled by the cell and it's going to stain the background. So we're going to end up with a red background, right? And then methylene blue is a basic stain that we're going to use today. So it has what charge?
Positive.
Positive. What's it going to stain? The cell. Right? So we're not ending up with purple here. It's not preschool. We're not mixing red and blue to get purple. Right? We're going to be staining two different components because of the two different charges. Very similar to what we're going to be doing next week with the capsule stain. Right? We're going to use two different charged dyes or reagents. So we're going to stain the cell and we're going to stain the background. So the two different procedures that we are going to do for lab today, one of them, we're going to heat fix our slides. Why do we heat them? What do we mean fix? What do we mean by fix?
Make them stick.
Make them stick, right? Because then we're going to dye them and wash off the dye. If we don't cause them to stick, they would wash off our slides, right? So that's called making a bacterial smear, where you spread them out on your slide and you heat fix them onto the slide. This is commonly done where you're going to use basic stains, okay, where we're going to actually have the stain go into the cells. When we do negative staining, when we use an acidic dye, we add the dye to the slide, then the organisms, and we spread them out. We do not heat them. We just let it dry. Okay. Heat fixing also kills them, but that's not why we do it, right? We do it to stick them to the slide. So the difference between these two procedures is that heating component. Does heat affect the size of a cell? Yes. What would heat do to it? Would it increase or decrease? Decrease. So that's why when you simple stain, because of how you prepare the slide for that heat fix step, you'll actually shrink those cells. So if you were to look at the same organism stained two different ways, and if you could measure the size, they're going to look smaller when you basic stain them than if you just did a negative stain. And that's because of the heating, right? Shrinks the cells. Is that my last one? I thought so.